Malaria. Malaria is caused by the pathogen Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae. These Plasmodium are a group of prototists. They are transferred or transmitted to humans through an insect vector known as the female Anopheles mosquito. Anopheles is a Greek word for useless. Global distribution would be in the tropics and subtropics, so it's an area where there is warm, moist climate. When we say vector, it is an organism that carries a disease from one person to another. Other than transmitting malaria or the plasmodium to humans through Anopheles mosquito, it could also be transferred to us through blood transfusion if the blood is unclean, unsterile needle, and mother to fetus. The incubation period for malaria would be from a week to a year, and it targets the red blood cells and then the liver and the brain. A person with malaria can feel high fever, anemia, nausea, headaches, muscle pain, shivering, sweating, and enlarged spleen.
The parasite Plasmodium falciparum is responsible for most of the world's malaria cases. It has evolved in concert with its human and mosquito hosts and displays a complex life cycle that alternates between the two. An infected female mosquito transfers the parasite to a human when she takes a blood meal. The parasite, in the form of sporozoites, is injected with her saliva, which contains an anticoagulant that promotes a steady blood flow. The circulatory system delivers the sporozoites to their target, the liver. A sporozoite enters a liver cell and begins to feed and grow. During the process, the parasite's nucleus divides many times, and then the nuclei bud off to produce thousands of new plasmodium cells, now in a form called merozoites. About a week later, the merozoites burst out of liver cells and enter the bloodstream. In the bloodstream, the merozoites infect red blood cells, where they again reproduce and burst out of the infected cells. The resulting merozoites can infect other red blood cells, yielding a cycle of infection and eruption. Malaria is characterized by repeated bouts of fever, each of which correlates with the synchronous release of merozoites from a population of red blood cells. Some of the infecting merozoites develop into sexual forms called gametocytes, which are either male or female. In the life cycle of Plasmodium falciparum, gametocytes become large oval structures within red blood cells. The gametocytes do not harm the human host, but when a mosquito takes the gametocytes into its gut during a blood meal, these cells initiate the next stage of the Plasmodium life cycle. In the mosquito's gut, the gametocytes escape from the red blood cells and develop into male and female sex cells called gametes. A male and a female gamete fuse to produce the first cell of the next generation, the zygote. The zygote matures into a motile cell called an ookinete and migrates to the wall of the mosquito's gut, where it develops into an oocyst. Inside the oocyst, cell division occurs to produce many new cells, again in the form of sporozoites. The oocyst ruptures and releases the sporozoites, which migrate to the salivary glands. From the salivary glands, the sporozoites can infect the next human host and repeat the Plasmodium life cycle.
This is how the normal red blood cell look like. It's biconcave, it's spherical, it's disc-like. There are no extra, the surface is smooth. But if you have an infected red blood cell because of malaria, there will be knobs in here, this yellow area, and once you cut the red blood cell, it will be containing malarial parasite inside. This is a micrograph. This one, for example, in here are normal red blood cells. But if there is a crescent banana or sausage shaped gametocyte, it'll look like this. This violet area in here, these violet areas in here in the red blood cell. And this one especially, there is a shown Plasmodium vivax that is growing right inside the blood cell. Obviously, this blood cell is totally different from the appearance of a normal one. There are many treatments that people is supposed to take, especially if you're traveling to an area where malaria is very high in rate. And the treatment or the medicine that will be taken should be administered before the travel, during the travel, and after you come from the trip. Okay? There are a lot listed. Actually, there are more compared to the ones found in the book, but we're not really required to specify which one is which. Okay? In the slide, you'll be seeing the term prophylactic at the bottom. Prophylactic drug is just a medicine used to prevent a disease. So that's the reason why you have to take an anti-malarial medicine or drug during, before, and after the trip. So for the first drug, we have quinine. It kills the malaria parasite. It's from the cinchona plant that you could find in here. Next one, chloroquine, used for interfering with the growth of parasites in the red blood cell of the human body. It also inhibits protein synthesis and spreading of parasite within our body. But there is a resistance that has developed in South America, Africa, and New Guinea. So we could say that chloroquine is not used there anymore. If you have been reading the news also, chloroquine has been all the rage because they think it's one of the possible medicine that could stop or hinder the coronavirus right now. Mefloquine. It is used in areas where resistance to chloroquine is evident, so that will be in the Latin Americas and such. It will be good for travelers as it is taken only weekly, not daily. But if you're gonna travel to the northern borders of Thailand, there is resistance to mefloquine. Therefore, you're not advised to drink this one. Proguanil. This one inhibits sexual reproduction of Plasmodium inside of a biting mosquito. It is often used together with chloroquine or atovaquone. Doxycycline is an antibacterial drug. It is good for last-minute travelers because the drug is started 1 to 2 days before traveling to an area where malaria transmission occurs. It is a very well tolerated medicine. It tends to be the least expensive anti-malarial drug. Prevention. We always say that prevention is better than cure, but what are the things that we could do so that malaria will not be affecting us? So for the first one, reduce the number of mosquitoes. When we say reduce the number of mosquitoes, you might think of insecticides right away, but there are several other things that you could do which is much more eco-friendly than an insecticide. First one, oil over surface of water. You could see in here, these are larvae, the wrigglers, the baby mosquitoes. They hang on the surface of the water so that they could breathe, right? So in order for them not to take in the air, what you would want to do is to add oil on top of it so that there will be no space for the siphon or the nose, quote unquote, of the wrigglers to cling onto. Next one, drain marshes, clear vegetation. Marshes and vegetation would be um, a patch of land wherein there is a stagnant water. And when you say stagnant water, it is a water that doesn't move. So if there is a body of water that is stagnant, it will be a breeding ground for mosquitoes. Next one, stocking ponds with fish. This is because there are fishes that eat wrigglers or mosquitoes. Some even say that you could use frogs, but if you have so much frogs, that will cause problem for the diversity of the ecosystem of the area if you use that. So it's better to have fishes instead. And lastly, there should be Bacillus thuringiensis to spray so that you could, prevent those regulars from even living. Next one, avoid being bitten by mosquitoes. They attack at dusk. Dusk is right before dark, like right before evening. So it's in between afternoon and total darkness of nighttime. That's when they attack a lot. And then you could see in here, these are not sponsored. These are what do you call this one? Insect repellent. So that this mosquito will be smelling the scent of these anti-repellents, and once they smell or detect this anti-repellents, they will not go and bite you because they could smell it from you. There is another version of this. They use patches instead. They put it on your clothes or cloth, and the effect is the same. This one in here is a mosquito net. It is a wire frame with cloth that is very small so that mosquitoes cannot enter. Then we have here citronella. If you have used Minyak citronella, that one is good for preventing mosquito bites as well. If you have citronella plants in the house, that could also prevent mosquitoes from going inside the building. Next, number 3, use drugs to prevent the parasite from infecting people. Similar to the drugs that we've already discussed before. There has been a lot of problems with fully eradicating or fully removing malaria in our system, and it is because of 5 reasons. One, there is increased drug resistant forms, as if you could have reflected in the slides, 3 slides before this one. There are a lot of drugs, right? But not all of them could be used at the same time, or there is no one specific only. It would depend on the area where you're going. Number 2, increase in cases caused by the Plasmodium falciparum. Number 3, difficulties in developing vaccine because it's difficult to detect a parasite if it's hiding inside the red blood cell. Number 4, increase in epidemics because of environmental and climatic changes. Number 5, migration of people where malaria is epidemic, meaning there are more people going into these places, and since there are more people, therefore there are more victims of this parasite or this protoctist. All these constraints are the ones that are listed by the World Health Organization. I did not make this one up. So how can we eradicate or remove malaria from its existence at least? 1, improve diagnosis of malaria. Number 2, improve supply of effective drugs or reduce the drug resistance of the parasite or the protoctist to the available drugs. Number 3, develop vaccines against different stages of the parasite's life cycle, which is kind of difficult because we don't yet know how to target it, but I hope they're in the process already. Number 4, international will to remove the burden of disease from the poorest parts of the world. Number 4 just means that all the countries that are involved in the United Nations would come together and have a solid plan in their own country on what they would do in order for malaria to not exist anymore. This is an example of the dipstick test for malaria. In here, you can see in letter A, urine dip, there is a stick dedicated to malaria. And this one will be incubated for 20 minutes. After 20 minutes, the results will be compared. If it only has one line, it means you don't have a baby. Just kidding. It means that you have or do not have malaria. But if you have 2 lines sticking out, it means that you're positive for malaria. This one is actually a fast test already. Unlike before, you have to collect the blood, let the blood go or undergo centrifugation first, and then look at the microscope, right? This one is way faster than that one. This is another type of dipstick test wherein if you have nothing, just like this, there's no line at all in the middle area or this left region, you are negative for the malaria. But if you have one, it means that you have the strain Plasmodium vivax. But in here in the third row, if you have two lines, it means that you have the Plasmodium falciparum strain.